Cell Processes by Sarov, Maxby, Daniel, and Basile. pH is a measure of acidity in a solution. A pH of 0 is very acidic and contains large amounts of H plus ions. A pH of 14 is very basic and contains large amounts of OH minus ions. 7 is considered neutral, and it's an equal amount of OH minus and H plus ions. Cells prefer neutral pHs because pH affects the structure and function of many different molecules inside of a cell. A buffer is what allows a cell to maintain constant pH by removing or adding H plus ions to even out the pH of the cell. Cells maintain a stable inside environment regardless of the outside conditions, and this is referred to as homeostasis. They also can do this with water levels inside of themselves. So, if a cell has a higher concentration of solutes inside itself, water will rush into the cell to try and balance out the concentration of both sides. This is referred to as a hypotonic solution, and we can see that the animal cell and the plant cell swell up due to the excessive amount of water entering them. An animal cell is referred to as lysed in this condition, and a plant cell is referred to as turgid in this condition. When the concentration of solutes is even on the outside and inside of the cell, it is referred to as an isotonic solution. In an isotonic solution, water continuously moves in and out of the cell, but this is considered normal because the cell's concentration on the inside and outside is the same, and the pressure is also maintained the same on the inside and outside. This is a normal condition for an animal cell, but for a plant cell, it is referred to as flaccid. In a hypertonic solution, the concentration of solutes outside of the cell is much higher. This causes water to rush out of the cell in order to balance out both sides evenly. In an animal cell, this solution is referred to as shriveled or crenated, and in a plant cell, it is referred to as plasmolyzed. How do cells maintain the homeostasis of glucose and temperature levels? In cells, they use active transport, which uses ATP to move glucose in and out of a cell through the transport proteins found in the membrane. We can see this happening here, and this allows the cell to constantly balance and make sure that it has just the right amount of glucose inside of itself. The human body also performs homeostasis when it comes to the internal body temperature. We can see this because if a human body is much colder, shivering, which is the rapid contracting of muscles, generates heat, which helps to warm your body up. If it's very hot, your body sweats, which allows it to cool down in a warm environment.
There are two processes of reproduction in the body, which are mitosis and meiosis. While these are similar, there are some main differences. Mitosis is asexual, and it ends with diploid cells. Diploid means that it ends with all of the DNA of the parent. This creates exact copies of cells and is useful for, say, replicating body cells, which contain 46 chromosomes. At the end, there are two cells. In meiosis, it's sexual reproduction, which means that the chromosomes will be different due to genetic variation. This will end with haploid cells, half the number of chromosomes, from the parent. Gametes have 23 chromosomes due to this, and at the end, there are 4 cells produced. So how does meiosis make genetic variation? This is achieved through crossing over. So, as you can see in this diagram, you have homologous chromosomes, or tetrads, as they are called after crossing over. They have exchanged a small part of DNA, as you see the dark chromosome has exchanged a piece with the light chromosome, and now they have different traits. So, once a sperm or egg cell meet, fertilization occurs. In the diagram, this little thing with a tail is the sperm cell, and the circle is the egg. At the end, they create the zygote, which is the finished sex cell, and will turn into an embryo. Chromosomes are distributed by random assortment. They're not sorted by any particular order, such as size or what they code for. If a cell doesn't get the right amount of DNA, that is called non-disjunction. Having odd numbers of chromosomes creates genetic disorders. So as you see, there are 4 in the original cell. It would be 2 and 2 if this were meiosis, but instead it's 3 and 1, which could cause problems within the body. And a gene mutation is simply a change in DNA. It could be an adding of a base pair, which codes for the trait, or it could simply be a shift in what the trait actually is later on.
So there are two types of movement of molecules in the body. The first one is passive transport, which is the movement of molecules that does not require energy. It is a movement from an area of high concentration to an area of low concentration, and it does not use energy. There are two types of passive transport. Osmosis is when only water is moved from an area of high concentration to low concentration, and diffusion is the movement of any other molecule. Active transport is like passive transport, but it requires energy and moves molecules from an area of low concentration to an area of high concentration. This can be of water or any other molecule, and big molecules are moved through transport proteins. Interphase is part of the cell cycle, and it includes G1, in which the cell grows, S phase, in which DNA replicates, and G2, in which the cell grows more. However, there is an outside stage, which is called G0, and is a stage in which the cell doesn't do anything and just ends up dying because it can't replicate. Mitosis is cell asexual reproduction, and it is broken into 4 stages. Prophase, in which the chromosomes condense, the spindle fibers move to opposite side, and the nucleus starts to dissolve. Metaphase is when the chromosomes line up in the middle, and all spindle fibers are attached to the centromeres. Anaphase is when the spindle fibers start pulling the sister chromatids to opposite side of the cell. And telophase is when the nucleus starts to reform and the interphase starts to happen again.
So, why do organisms perform mitosis and meiosis? Well, in mitosis, they replicate and really fast, and mitosis is for growing, healing, all of that. So, for example, if you got a cut on your hand, it would heal back, and that's an example of your skin or your skin cells performing mitosis. And in meiosis, the purpose of meiosis is to reproduce and pass down genes, and that allows for improvement and evolution in the species.